Hello, welcome to Biology, the scientific study of life, the genetic basis of development. From single cell to multicellular organism. This chapter applies much of what we have learned about molecular cells and organs to one of biologists' most important questions: how a complex multicellular organism develops from a single cell. The application of genetic analysis and DNA technology to the study of development has revolutionized the fields. The same way that researchers have used mutations to deduce pathways of cellular metabolism, they now use mutations to dissect developmental pathways. In one striking example, Swiss researchers demonstrated in 1995 that a particular gene functions as a master switch that triggers development of the eye in Drosophila. The scanning electromicrograph in this figure, notice that shows part of the head of an abnormal fly that has a small extra eye on each antenna. In this fly, notice that master gene triggering eye development was expressed in an abnormal body location, causing extra eyes as similar gene activities eye development in mice and other mammals. In fact, developmental biologists are discovering remarkable similarities in the mechanisms that form diverse organisms. The scientific study of development got underway about 150 years ago, around the same time as genetics. But for decades, the two disciplines proceeded along mostly separate paths. Developmental biologists focused on embryology, the study of the stages of development leading from a fertilized egg to a fully formed organism. They studied animals that lays their eggs in water, including marine invertebrates and freshwater vertebrates, amphibians, such as frogs. By studying these and other animals as well as plants, biologists worked out a description of animal development at plant development at the macroscopic and microscopic levels. In recent years, scientists have applied the concept and tools of molecular genetics to the study of developmental biology with remarkable fruit fly results. In this chapter, we will introduce some of the basic mechanisms that control development in animals and plants, focusing on what has been learned from molecular and genetic studies. Okay. Notice that also the primary researcher goal is to understand the broad biological principle. The organism chosen for study is called a model organism. Researchers select model organisms that lend themselves to the study of a particular question, are representative of a larger group, and are easy to grow in the lab. The first one is model organisms of genetic studies of development. The first one, Drosophila melanogaster fruit fly. Researchers can draw on a wealth of information about the fruit fly Drosophila melanogaster, after referred to simply as Drosophila. One of the most important model organism in developmental genetics, first chosen as model organism by the pioneering genetists T.H. Morgan in the early 19th century. Drosophila has been studied intensively by generations of geneticists since then, small and easily grown in the laboratory. Drosophila has a generation time of only 2 weeks. It produces many offsprings, and the embryos develop outside the mother's body. Both assists for developmental studies. Sequencing of the Drosophila genome was completed in 2021. It has 180 by 10 to the power 6 base per 180 million bases MB short term. and contains about 13,070 genes. Although early development of fruit flies is at least superficially quite different from that of many other animals, search on Drosophila development has yielded deep insights into a basic principle of animal development. The second one is notice that is canorophyba or nematode canor habidite elegans. The short term is nematode. The soil-dwelling nematode Canor habiditiis elegans, short term C. elegans, is easily grown in the laboratory in petri dishes. It is about 1 millimeter long, has a simple transparent body with only a few types of cells, and grows from zygote to mature adult in only 3.5 days. The nematode genome is 97 MB long and contains an estimated 19,000 gene. Most individuals are hermaphrodites, producing both eggs and sperm. Hermaphrodites are convenient for genetic studies because recessive mutations are easy to detect. If a worm with the wild type phenotype self-fertilizes and 1/4 of its offspring have a mutant phenotype homozygous for a recessive allele, then the parent must be heterozygous for the recessive mutant allele, even if homozygote with the recessive mutant do not reproduce. The mutation can be maintained in the heterozygotes. Another advantage of C. elegans is that every adult hermaphrodites. Another advantage of of C. elegans is that every adult hermaphrodites has exactly 958 somatic cells, which arise from the zygote in virtually the same way for every individual. Using a microscope to follow all the cell division starting immediately after a zygote forms, biologists have been able to reconstruct the interior ancestry of every cell in the adult body. Third one is most muscular mouse. Notice that. Okay. Among vertebrates, two in particular lend themselves to the genetic analysis of development: the mouse and the zebrafish. The mouse, muscular, has a long history as a mammalian model, and much is known about its biology. The mouse genome is about 2,600 MB long, with about 25,000 genes, roughly the same as the human genome. Researchers are now adept at manipulating mouse genes to make transgenic mice and mice in which particular genes are knocked out by mutation. However, mice have a generation time of about 9 weeks, and their embryos develop in the mother's uterus. Hidden from view, both disadvantages for developmental studies. Fourth one is Daniorario zebrafish. Many of the disadvantages of the mouse as a vertebrate model are absent in the zebrafish Daniorario. These small fish, 2 to 4 centimeter long, are easy to breed in the laboratory, and the transparent embryo develop outside the mother's body. Although the generation time is relatively long, 2 to 4 months, early development proceeds quickly. By 24 hours after fertilization, most of the tissues and rudiments of the organs have formed, by 2 days, a tiny fish hatches out of the egg case. The zebrafish genome is estimated to be 1,600 MB long, is still being mapped and sequenced, but researchers have already identified many genes involved in this animal's development. Fourth one is Arabidopsis thaliana, or common walrus. For studying the molecular genetics of plant development, researchers often use a small flowering plant in the master family called Arabidopsis thaliana, for simply Arabidopsis. One of these plants can grow in a test tube and produce thousands of progeny after 8 to 10 weeks. As 10 Mendel's pea plants, each flower make both eggs and sperm for research on gene function. Scientists can make transgenic Arabidopsis plants. Notice this one. We will compare with some other plant species. Arabidopsis has a relatively small genome, about 180 MB, which contains an estimated 500 genes. Okay.